Good morning, students. Welcome back to Biology class, Chapter 9, Part 3, Strategies for Enhancement in Food Production. Okay, today's class we are going to discuss about the plant breeding. Plants are the most important organisms on this planet. Plants are the producers. Almost 85% of the world's food consumed by humans is plant-based food. Plants grown for food are called food crops. Food crops are highly dependent on water availability, soil, climatic conditions. Sometimes, due to environmental conditions like drought, the crops do not produce sufficient food or the crops fail. This leads to acute shortage of food in that region. It is called as famine. If the food production does not increases, it will cause food crisis. As the land for agriculture is very limited, it is necessary to improve the productivity of the crops per acre. As the population increases, food production must also increase. If the food production does not increase, it will cause food crisis. As the land for agriculture is very limited, it is necessary to improve the productivity of the crops per acre. By implementing animal breeding, we were able to improve the quality and productivity of farm animals. Similarly, by plant breeding is defined as a science of changing the genetic traits of plants in order to create desired plant types that are better suited for cultivation, given better yields, and have good resistance to diseases. Traditional farming can only yield a limited biomass as for food for humans and animals. Better management practices and increase in acre can increase yield, but very limit. Plant breeding is a technology has helped to increase the yield in a large extent. In our country, Green Revolution was responsible for the national requirement in the food production. Green Revolution was dependent to a large extent on plant breeding techniques for development of high-yielding and disease-resistant varieties in wheat, rice, maize, etc. Plant breeding is the purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation, give better yield and disease resistant. Conventional plant breeding has been practiced for thousands of years since the beginning of human civilization. Recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. Many present-day crops are the result of domestication in ancient times. Today, all our major food crops are derived from domesticated varieties. Classical plant breeding involves crossing or the hybridization of pure lines followed by artificial selection to produce plants with the desirable traits of higher yield, nutrition, and resistance to the diseases. Plant breeding is not a new technology. It is very old one. Earlier, man used to deliberately crossbreed closely related plant varieties to produce a new plant of desired trait. Nowadays, breeders try to incorporate the following desirable traits in an improved variety, that is increased crop yield and improved quality, increased tolerance to the environmental stresses like salinity, temperature, drought, resistance to the pathogens like virus, fungi, bacteria, and increased tolerance to insect pest. Okay, now this process of breeding closely related individuals that are genetically different to produce a hybrid offspring is known as hybridization. Plant breeding, one important point, it is the hybridization. Two related individuals combine to form a hybrid offspring. Gregor Mendel and Charles Darwin effectively demonstrated hybridization within a plant species. The plant breeding program carried out in a systematic way in the worldwide. The first point, collection of variability. Second point, evaluation and selection of parents. Third point, cross hybridization among the selected parents. Fourth point, selection and testing of superior recombinants. Fifth point, testing, release and commercialization of a new cultivators. Okay, now first we will go through the first point, that is the collection of variability. Genetic variability is the root of any breeding program. In many crops, pre-existing genetic variability is available from the wild relatives of the crop. Collection and preservation of all the different wild varieties, species and relatives of the cultivated species, evaluation by their characteristics is a prerequisite for effective exploitation of natural genes available in the populations. We can select the less drought resistance or good drought resistance, more yield, good quality. Some give high yield, some give less yield, no resistance to drought, some drought resistance. Best required traits combine to form a new plant. From the year 1971, under the guidance of International Board of Plant Genetic Resources, germplasm collection contains more than 100 varieties. In India, there are 2 germplasm collection centers are there. International Crops Research Institute for Semi-Arid Topic at Hyderabad. Second one, National Bureau of Plant Genetic Resources at New Delhi. In that collection of variability, the entire collection of plants or seeds having all the diverse alleles for all genes in a given crop is called germplasm collection. Collected ones and getting a new plant. Now the second point, evaluation and selection of parents. The germplasm evaluated so as to identify plants with a desirable combination of characters. The selected plants are multiplied and used in the process of hybridization. Pure lines are created wherever desirable and possible. The third point, cross hybridization among the selected parents. Okay, the desired characters have very often to be combined from two different plants, two different parental plant. For example, high protein quality of one parent may not be combined with the disease resistance from another parent. This is possible by cross hybridizing the two parents to produce hybrids. that genetically combine the desired characters in one plant. This is very time consuming and tedious process since the pollen grains from the desirable plant chosen as a male parent have to be collected and placed on the stigma of the flowers selected as female parent. Also, it is not necessary that the hybrids to combine the desirable characters. Usually, one in few hundred to 1,000 crosses shows the desirable combination. Okay, hybridization. Artificial hybridization is the production of hybridization by cross-pollinating two separate plants under controlled conditions and the supervision of the plant breeder. The process of artificial hybridization in plants involves various steps. First point, selection of parents with desired characters. All the desired characters should be present in the parents which are required in the new variety. Second point, parent with desired character. The third point, emasculation. What is emasculation? The female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is called emasculation. Removal of the anther from the bisexual flower. Then, next point, the emasculated flowers. Remove the anther. Then the emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent the contamination of its stigma with the unwanted pollen. This process is called bagging. So the artificial cross hybridization, the first point, selection of the parents with the desired characters. Second point, emasculation. Third point, bagging. Then, when the stigma of bagged flowers attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma and the flowers are re-bagged and the fruits to allowed to grow. Okay, the emasculated flowers, first collection of the pollen grains. Artificial pollination. When the stigma of the emasculated flower matures, the polythene bag is removed and with the help of a brush, fresh pollens from selected parents is dusted over the receptive stigma of the females. Then next point, it is the tagging. Okay, artificial hybridization. Next point, it is the tagging. A tag is with the relevant information attached to the plant. The tag bears the information like date of emasculation, date of crossing, and the details of male and female plants, etc. Next, desirable combination of the variation. Seeds obtained after crossing are sown any evaluated for desirable and undesirable traits and are screened properly. So the limitations already we saw that when the hybridization, the first, this is a very time consuming and tedious process for the pollen grains to mature. Then the second point, the cross hybrids may not combine the desirable characters. Usually one in few hundreds to 1,000 crosses shows the desirable combination. Next point is the selection and testing of a superior recombinants. This step consists of selecting among the progeny of the hybrids those plants that have the desired character combination. The selection process is crucial to the success of the breeding objectives and requires carefully and scientific evaluation of the progeny. This step yields the plant that are superior to both of the parents. Very often, more than one superior progeny plant may become available. These are self-pollinated for several generations till they reach a state of uniformity or the homozygosity, so that the characters will not segregate in the progeny. Cultivar is a plant with the desirable characteristics that can be maintained by propagation. The cultivar are grown in research field and their performance is recorded under ideal fertilizer application, irrigation and other crop management places. In India, Indian Council of Agricultural Research at New Delhi carries out these evaluations. After evaluation, the plants are again tested in farmers' field for at least 3 growing seasons. So next point, it is the testing, release and commercialization of the new cultivar. Cultivar means a plant with desirable characteristics that can be maintained by propagation. The newly selected lines are evaluated for their yield and other agronomic traits of, means that how the soil, traits of quality, disease resistance, etc. This evaluation is done by growing these in the research field and recording their performance. Under ideal fertilizer application, irrigation and other crop management practices, the evaluation in research field is followed by testing the materials in the farmer's field for at least 3 growing seasons at several locations in the country, representing all the agroclimatic zones where the crop is usually grown. The material is evaluated in comparison to the best available local crop cultivar. A check for the reference of the cultivar. The seeds of the approved cultivars and multiplied by growing them in suitable conditions, seed size, moisture, quality, color. Such tested seeds are called as certified seeds. Now the seeds can be distributed to farms and licensed to the seed sellers. Thank you.